A very good evening and welcome to one and all present here for this webinar on DENTOPHARM 2020 on sterile inflammation by implants and CAR T-cell cancer therapy. I'm Dr. Ramya, part of Dental Education Unit and Senior Lecturer at Sri Balaji Mittal College and Hospital. We'll be highlighting you about this program. I would like to welcome our chairperson, Dr. Sri Nisha ma'am, Registrar Dr. Bhuminathan sir, Principal Dr. Jimson sir, and Dr. Sumati Jones ma'am, Head of the Department of Pharmacology at Sri Balaji Dental College and Hospital. Over to Sumati ma'am to share a few words on today's webinar.
Good afternoon to one and all present here. DentoForm is a common platform wherein researchers from multidisciplinary area will explore their views, ideas, and their expertise in their respective research field. So this will serve as a common platform for all the researchers, not specific to any field. So our speaker, Dr. Pankaj, is well-known researcher in the inflammation and immunological area. So I would like to call upon Dr. Pankaj sir to present his speech.
This program throws a knowledge to the youngsters to obtain a conceptual understanding to the research, latest drug targets, drug discovery, drug nanotechnology, medicine, pharmacological and toxicological aspects, chemotherapy, biomedical analysis. A little announcement on housekeeping before we get started. So if you have any queries during the presentation, please type them in the chat box or raise your hand. I'll bring them up after the presentation and we will have time for questions at the end. I warmly welcome guest speaker of today's webinar, Dr. Pankaj Mishra sir. He did his master's doctorate at Center of Biotechnology, Anna University, Chennai. He was faculty at Department of Biotechnology at Birla Institute of Technology, Mesra, and then he moved to North America for postdoctoral research. He did his postdoctoral research at University of Toledo and Ohio State University, and then worked as an associate at Center for Immunity and Inflammation during pandemic. He started working at Public Health Research Institute, Rutgers, on COVID-19. Rutgers University as research associate and later became research scientist. His research primarily focuses on immunology and understanding the mechanism of inflammation for type 2 immune response caused by parasite and solid particle mediated sterile inflammation. He has also published 19 research articles in various high impact journals. He is one of the pioneers in understanding the mechanism of inflammation caused by sterile particles used in orthopedic implants, which was published in Nature Material website. He is currently a principal scientist at Merck Research Laboratories, where he is working on development of CAR T cell therapy for cancer. Now, without any further delay, we will time over to the speaker, Dr. Pankaj Mishra, sir.
Hi, good morning from here and good evening over there at Chennai. Welcome to everybody. I'm so excited to participate in this DentoPharm 2022. I'd like to thank the principal, Dr. S. Jimson, for inviting me to speak in this seminar, and Dr. Sumati Jones, who contacted me through her old friends, Dr. Madhuri, and that is how they contacted me for participating in this particular DentoPharm conference. I would also like to thank other organizing committee and Dr. Ramya for a very good introduction, and Mahima for helping me out to set up all these slides. Okay, so as you know, the title of my talk is today Sterile Inflammation by Orthopedic Implant and CAR T cell for cancer therapy. So it has got two topics. So first, I will finish the first part, which will cover around 70 to 80% of my talk, and we'll have a break and question answer session. We'll finish that, and then we'll go to the CAR T cell therapy portion. Okay, so I, like, when I was told, like, it is for biology dental college, so I thought, like, or something. I work on various model of inflammation, and by profession I'm a immunologist. I thought probably, like, sterile inflammation is something which will be very interesting to dental student. Also, when I was presenting this particular work in different conference like American Association of Immunology or inflammation seminar, I used to see a lot of dentists come and they ask questions. And even after my paper, a lot of dentists, even some good dentists from Italy and all, like, they write questions and I need to answer. So I thought, like, probably this will be something very interesting to all the dental students and others who are associated with this dentist school. So before I start, I'll have to do self-declaration that I'll be presenting this work based on my own research work and other published research article. This presentation will not include any of the research that I might have been worked or working associated with any of the biotech or pharma industry that I may have collaborated or worked directly or as a consultant. This talk is purely for academic purpose and for teaching medical students. So then the first slide. So we, like, all these projects started with my interaction with some of the orthopedicians back in New Jersey Medical School. uh, there were very good orthopedicians, and they don't know, they do a lot of surgery, and they do see a lot of they need to do a lot of revision surgery, and they wanted to understand. So it all started with a meeting, sitting front to front, and we all started these. So no one knows, like, what happens, what whether the solid microparticles which is released in orthopedic implant, do they cause inflammation, what they do, no one knows exactly. There was no thorough study on these. So first of all, I'll try to explain you what is sterile inflammation. We all know, like, there are inflammation happens whenever there is an infection. Like, you can get virus infection, you can get infection with bacteria, you can get infection with fungi, you can get infection with parasite infection, and all these things cause inflammation. And there is an immune response, because we need to have an immune response that will give us protection. So whenever there is no, what is sterile inflammation? Sterile inflammation is nothing but it is inflammation when it happens without any infection. So without infection, how it will happen? Does the immune response recognize some non-infectious foreign substance? So what are the causative for sterile inflammation? One thing, it could be allergen. Allergen are non-infectious, and the allergen-related inflammation is so common back in here in North America. Then tissue injury, like, say for example, there is some wounding. Of course, like, co-infection can happen. It happens that with the tissue injury, there is a sterile inflammation, but it's there is also, like, always there is co-infection. Then these days, like, diesel exhaust particles, back in Chennai when I was there, I remember, like, driving in the bike on the road, you get a lot of, like, all this petroleum product and a lot of exhaust particle. Delhi is so polluted, so those diesel exhaust particle directly you can breathe, and it can go to your lung, and it can also cause some inflammation, and it can probably damage the lung. So same way, like, implant materials, they can also cause inflammation, and that is what is called sterile inflammation. Scientists have worked, like, a lot of things on infectious disease like bacterial and virus, and also for parasite, and they have a very good understanding of the mechanism of inflammation. So here in this thing, we wanted to know what happens with the solid wear particle. And it happens that some of the orthopedic wear material even are used in dental surgery. So that you'll be knowing better than me. So just to briefly, in a bacterial or viral inflammation or fungal inflammation, usually there is a Th1 or Th17 inflammation happen. I'll explain you sometime later what is it. And with the parasite, basically there is a Th2 type of inflammation, and macrophage plays a major role. uh, in a Th1 or Th17, basically you see M1 macrophages, and in a Th2 inflammation, you see M2 macrophage. And we'll talk a little more detail in next slides. So yeah, looking at the dental, I was looking at, so they do definitely titanium. They use some of the implant zirconium, polyethylene, and these all. But there is a difference between dental implant and orthopedic implant. Orthopedic implants are used for mostly knee surgery. Here people don't want to get pain. Most of the people prefer to do the knee surgery, and they remove their knee and they put a cobalt chrome cuff, and but what happens is when they walk, actually slowly small particles come out of that, and then that causes inflammation, and some of the inflammation becomes severe, that if they're not taking anti-inflammatory drugs, or depends on their age and sex and their other conditions and also co-infection, usually there is a lot of time they need to do revision surgery, and not only once. There are some cases where they need to do many times. So I wonder, in the teeth, particularly doing the dental surgery, I don't know how the particles will be coming. You will be knowing better. But usually, in the medical terminology, it has been told, these are all inert particle, and these are safe to use. But if the particles are coming out of it and it is releasing and it is going to the body is going to respond to you. It body is going to take it as a foreign particles. So, talking about immunology, what happens when there is an infection? What happens? It is always like in our body system. It has been designed in such a way that any kind of thing happen, we can survive. And our with the evolution, our immune response has been so well trained. It is just like a program. See, if you want to protect India, we need to have border security in all the border. There are strategy. First strategy, there is border security force. Then second strategy, maybe we want to use aeroplane, or maybe you want to use aircraft, or maybe you want to use drones. All these kind of things happen inside the body also. So these are some of the things. And see over here, neutrophil, eosinophil, basophil, natural killer cell, macrophage, dendritic cell. So these are all innate cells. So whenever there is a, say for example, infection, let's take an example of infection. If you get infection, all these innate cells are the one which try to go fast, and they'll try, especially macrophage, it'll go and grab, it'll eat all the things, whatever you will see. They'll be traveling through your blood circulation. The macrophage will be all going all around your body, and the moment it sees something foreign, it will grab it, actually it will eat it. And even your own body has in our body is designed such a way that a lot of inflammation happens. Always a lot of cells are dying, and who clears those dead cells? Those macrophages. So macrophage, the one which goes and take it, then it'll innate immune response. So first 48 hours to 72 hours, you see a strong innate immune response. Then they process it. So these are our first defense mechanism, or you call first border security force for keeping our health safe. And they do in a very programming way. There's a program, there's a way, and they try to understand. They really don't go and interfere with your own body. So they have an understanding, and there are many mechanisms. The way they do is that there is a receptor ligand, They try to recognize whether it is they can eat or not. If it is a smaller particle, like say for example, if it is a virus, a bacteria, macrophages are very big in size. They can immediately go and eat it actually. Okay, and then once they eat, they'll process it and they'll do antigen presentation. They also go to the lymph node. The nearby is the lymph node, or they'll go to the spleen, and that is a place where a lot of B cells and T cells are there. And then they exactly map those B cell and T cell selections, and they map them to map antibody against those foreign particle, like so that a B cell will go and bind to the antigen and it will be almost arresting. And then again, once they arrest everything, T cell will go and there are killer T cell, they'll clear it, everything. Same way there is natural killer cells and gamma delta T cells. They are part of both innate response and adaptive response. So natural killer T cells are again they're kind of innate response, and gamma delta T cells also they can be both part of innate and adaptive immune response. So again, talking about these are classical, in immunology, everyone knows if you are infected with a virus or intracellular bacteria, the response happened is Th1 response. It is also called type 1 inflammation. And mostly it happens by cytokines are nothing but it's just like you are firing gun, and one interferon gamma goes, it's just like a signaling network, interferon gamma binds to the interferon gamma receptor, and then whole cascade of gene will get expressed downstream of that, so that they'll produce porphyrin, they'll produce a lot of enzymes, proteases, a lot of things happen downstream of that, and it happens in a programmatic way. So the immune response need to identify structurally whether is it an intracellular organism. If it is intracellular, mostly they try to produce a Th1 type of response, and that is dominated by interferon gamma and TNF alpha. There are some other cytokine also, but just to make it simple, Th1 response, which happens in bacterial or viral infection, is mostly dominated by cytokines like interferon gamma and TNF alpha. But when it comes to extracellular particle and sterile solid microparticle, you know, then there is a different type of exactly different type of response happen, that is called Th2 inflammation, and that is by mostly interleukin 4 and interleukin 5 cytokines, and of course there is interleukin 13 and interleukin 33. And even allergens, some of the allergen produce Th2 type of immune response, and sterile inflammation, like with solid microparticle, you get the Th2 inflammation. For fungi, there is some of the fungi are also extracellular, but instead of producing Th2, they produce Th17 inflammation. That is mostly by interleukin 17 and interleukin 22. So today we'll be talking mostly about the middle part, the Th2 inflammation, caused by sterile solid microparticle. This is as I used to work. This is from one of my review paper actually in Nature Mucosal Immunology. What happens with the helminth parasite infection? Because these are gut helminth parasites. They go to the gut, and what they do is that they damage some of the epithelial cell. You see, just the damaging, because the parasite will be dancing inside your tummy, and it will slowly, very mildly it will damage. And some of the parasite, even from your skin, they can enter, they find the way, you never get pain, right? They can go to the lung, and first they go to the lung, and then from the lung, they go to the gastrointestinal system. They can map pore, tiny pore, and they can go, and they do tissue damage with their travel. And with the tissue damage, what happens is cytokines like interleukin 25, interleukin 33, and there is a TSLP. That is the three cytokines which come, and that kickstart the whole Th2 inflammation. This is what is the latest in our understanding of the Th2 inflammation. And then these tissue damage, the damage that happens, the damage is map alarm. It is just like, hey, there is something going on. Someone is damaging us. We need to do something. And these three effector cytokine, IL-25, IL-33, and TSLP, they come, and that makes a lot of innate cells come, like inner cells like macrophage, basophil, eosinophil, mast cell, dendritic cell. These are all inner cells. They come very quick, actually. Within 3 to 4 hours of damage, they'll come, and then they produce different cytokines. And then, there is something called recently, innate lymphoid cells. That is also something like innate cells. They are small in size, and this is a new discovery, actually, back 3 to 4 years before that. ILC2, innate lymphoid cell type 2, they produce interleukin 33, 13, and then they can change macrophage to or M2 macrophage because of parasite. M2 macrophages come. Then CD4 cells produce interleukin 4, interleukin 5, and interleukin 13. And then once they want to reduce, once inflammation happens, then there has to be stop the inflammation. So for that, Treg cell comes, which produce FOXP3, and there is interleukin 10 is an anti-inflammatory cytokine. So then slowly, it neutralizes. So this is what our understanding from the helminth. Then we'll talk about here a lot about M2 macrophage, because with the sterile inflammation, what we see is that the M2 macrophage plays a critical role. You see there are two types of macrophage. One in the green, the top one, that is the M1 macrophage. This is a classical macrophage. Classical macrophage they call, because initially our macrophage and our understanding was all with M1 macrophage. Traditionally, whenever you get an infection with bacteria or virus, you get the M1 macrophage, and they produce TNF alpha, they produce interferon gamma, interleukin 2, and these are pro-inflammatory. Even autoimmune diseases, you see a lot of M1 macrophages, actually. But however, with a helminth parasite or sterile inflammation, what we see is that you get M2 macrophages. They are stimulated by interleukin 13 or interleukin 4, and they put type of cytokine. And in the literature, it has been taken as anti-inflammatory. But when too much of M2 macrophages come, and based on the microenvironment, they could be even inflammatory. That is what my research says, and we'll see slide by slide. Okay, so here my aim was to tell you, there are two type of macrophage. One is M1, and the other one is M2. And M2 is very critical in sterile inflammation, and we'll learn more about it. So our specific aim for this project was to examine the nature of immune response generated by wet debris particles released from prosthetic devices used in hip and knee joint replacement. When we get these initially, I talked to the orthopedic implant, and we thought they may not be producing inflammation. So we first asked the question: do wet debris particle induce an inflammatory response? And there are, in the literature, people have done some in vitro work, and they have shown Th1 type of response, they have shown Th17 type of response, they have shown Th2 type of response, but it's not clear. And there are some written it doesn't cause even inflammation. It depends on the models, actually. So being an immunologist, it was kind of a challenge, actually. So we wanted to ask questions: what type of immune response generated? And one of the major things that we wanted to, if there is a immune response, what is the mechanism, and how they're doing inflammation? So with this study, you'll get an idea about what are the different mechanism and what kind of approach you do to understand truly from basics when not much is known. So those kind of information you will get from this seminar. So I published a paper first, in Journal of Immunology. First thing what we did is that we could get titanium particle, because other orthopedic implant material we didn't have access. There is no market where you can buy, actually. So we are fortunate, we could buy from titanium microparticle. But the challenge was that, being an immunologist, I ask questions. People, just the particle you take, that will have LPS. LPS, what is LPS? Lipopolysaccharide. And LPS is all the bacteria, and LPS is one of the major stimulant. If you put LPS into the body itself, there will be inflammation, because it will consider LPS is major part of the cell membrane of the bacteria. So that will give a TH1 information. So one of our major thing we did is that we had a collaborator in Chicago who is a material engineer, Dr. Halaf, and also he's an orthopedic expert. And we asked him to, can you make the material sterile for us and measure the size? And we want to study the microparticle. Those are bigger in size actually. And we got that. There is a big procedure. They need to treat with acid. They have a but they need to autoclave for, like, instead of for 1 hour, they need to autoclave for 12 hours, and that is a particular way. And also the what is the structure they check with their different scaling electron microscope and law assay, measure the size. The particle that we get is like ranging from 0.8 to 88 micron. So some of the particle they can immune response can take it, and some of the particles are really bigger in size. So we inject it to the mice, and then after 48 hours, we examine what's happening. Is there any innate inflammation? Because within 48 hours range, you will get innate inflammation. And what we see is that there is increase of neutrophil, there is increase in eosinophil, and there is increase in M2 macrophage. How do you do this? You do this by flow cytometer. I'll explain you how we analyze this. So we inject the mice with these particles, and then we do a peritoneal labase. So we get the peritoneal fluid, and those peritoneal fluid we run by flow cytometry, and we identify based on the marker what is there. We identify and we tell, hey, this many percentage are neutrophil, this much percentage are eosinophil, and this much are M2 macrophages. Then we do check their cytokine gene expression. You see, this is this what do you say, interleukin 4, interleukin 5, interleukin 13, interleukin 25, interleukin 33. These are the marker of TS2 inflammation, and we do see increase in these genes, and that's confirmed that this is something giving like some kind of an allergen response or type 2 inflammation. And we don't see much changes in interferon gamma, IL-12 or TH17. These are all TH1 and TH17 inflammation. For M2 macrophage, we see that these are the marker of M2 macrophages. Listen it carefully, because in next of my slides, all these genes will be there. So ARC1, it's a arginase 1, RELM alpha, and YMON. These are all markers. These genes are expressed, then it become M2 macrophages actually. So that M2 macrophage produce a lot of arginase, and with that they can do wound healing. They can do a lot of things actually. And in our case, they'll give inflammation, and I'll show you. That is what we try to understand how it is doing actually. And INUS is the marker for M1 macrophage. So you need to remember, I'll be in most of my slide, I'll show you the gene expression data. So what is the marker of M2 macrophage? It will be ARC1, RELM alpha, and YM1. If you see the increase in these three genes, then there is M2 macrophage. And what is the marker for TS2 inflammation? It's interleukin 4, interleukin 5, interleukin 13, interleukin 25, and interleukin 33. And also in the preliminary experiment, we put titanium with albumin, and we see their serum. We see increase in IgG1 and IgE. And then we thought so thou or Dr. Halap makes a sterile particle and sent us, but still we are not sure. There might be some LPS, because low level of LPS can also cause some kind of type 2 inflammation by some investigator studied. So in order to make that, we injected these particles to a TLR4 knockout mice. So what is TLR4? It is toll-like receptor. So basically, bacteria will have LPS, lipopolysaccharide, and if lipopolysaccharide will be there, they through the TLR4 pathway they do. So we are the first one to say that, hey, look, these particles make an inflammation, and looks like it's a type 2 inflammation, and it is independent of toll-like receptor 4. This was the first time we published, and this is what made the attention. Hey, looks like the sterile inflammation is something type 2 inflammation, and it is independent of LPS. So most of my study, I have used flow cytometer, and just as a lot of students are there, I'll give you a little basics actually. So what you do is that once you get the cells, actually you prepare the cells, you put the antibody to the cells, mixtures of cell will be there. If there is marker, so every cell, say for example macrophage will have F480 on their surface. They'll be displaying just like their hand, they'll be displaying on their surface. So the antibody will go and bind there. The antibody is bind to some of the fluorescent protein. And then what happens is in a flow cytometer, basically it will come cell by cell, and there will be a optic laser, light will fall, and then it will tell exactly, the detector will be able to say, hey, this is F480 positive. So then that's macrophage. Is it a M1 macrophage or M2 macrophage? If CD206, there is another marker for M2 macrophage. If it is F480 and CD206 positive, hey, this is M2 macrophage. So these results are very robust, and flow cytometer is so critical for understanding deeply immunology. So most of my next slide, what I'll see, there will be a lot of flow data. So don't get confused. This is the technique flow cytometer used to find the markers for different immune cells. Oh, yes. So this is from my colleagues, Nadim Halaf. So he analyzed the particle, the here, cobalt chrome particles actually. So it is in between 0.3 micron to 100 micron size. So some of the particles are small, and you'll see, I'll show you, small particles will be taken by the macrophage immediately within 30 minutes. Macrophages are very sensitive actually. The moment they see something garbage are there, they just want to eat it, and they may die, but they eat. This cobalt chrome is just as nothing, but it is just like a bullet, just like a gunshot, right? These are solid particle. Macrophages eat bacteria and virus, but what happens if a macrophage eat particles? Maybe they'll die. We'll see what's happening. So, and the right side is this is another thing, you do electron microscopy, wherein you take different thing and you find the range of their size. Actually, it confirms that we have a sufficient number of more of microparticles are there, and some small size particles are also there. And after autoclaving, we have but the structure is there is an integral structure, and this is what you do. So first thing we try to study, because we saw TS2 inflammation. So you see, do you know alum? Usually people use alum is one of the adjuvants. So we want to see whether does these particles has some kind of adjuvant effect. So in order to do that, you can do simply, but in order to do really a more immunologically correct way, you take a DO 11.10 transgenic mice. What is this DO 11.10 transgenic mice? Transgenic mice means the T cell will, here in these mice, the T cell will express over specific TCR. That means if you put over albumin, the T cell will increase. So what we do in this experiment is, I'm trying to explain you so that a student can get it. So you first get all the spleen, lymph node, everything, make a preparation of the cells, immune cells, and then you pass through a column wherein there will be magnetic bead with the antibody to CD4. So they will get separated, and then once you elute, you get a pure T cell. You see, this is flow cytometer, one of the example of the flow cytometer, and then we get around 90%, 90 to 95% pure T cells, and then we label the T cell with a color called CFAC. Okay, CFAC will stain it to red actually, and what happens then when you inject them, let me show the next. So we inject these to a normal mice, Balsi mice. So these are the Balsi mice at T cell, but we want to study what's happening exactly over here. So this is a T cell which has got a over-specific TCR, what you see in green, and these are labeled with CFAC. So what happens? This is the OVA peptide, because this will get stimulated with the OVA peptide. So you either give cobalt chrome or you give alum, or you don't give cobalt chrome, and you see what's happening to this cell. So if the good thing is that you can track by flow cytometer, if it grows, if there is a proliferation, the CFSE will keep on diluting, because initially, like, it is just like, the color will get faded. And you see, this is the high color, the CFSE level is so high, and this is a small population. This is how many generation, how many cell division has taken place. All these things you can come to know actually. So this is what something we did. And in fact, like, as you see over here, um, with the just with ovapeptide, you see the end, like, how much cells are not produced. These are non-dividing cells. The first pic what you see over on the right side, these are non-dividing cell. Once they are dividing with the ovapeptide itself, like, there are some T cells are increase, some divide actually, but not much. But with the covaltrum, here 38.9, it comes down to 10.3. With alum it is almost similar, like, almost close to 10. So these suggest this is a very classical immunologic experiment by which you can say, hey, this is a adjuvant effect. Then alum is known to give a type 2 immune response. So we wanted to study, like, what kind of cytokine they're secreting. We did the ELISpot assay. ELISpot assay is like, ELISA, but ELISA you check in cytokine, but these are all like, how much secreting cells are there. Exactly the number of cells which are secreting interleukin 4 and interferon gamma. So in the black color, what you see, like, these are interleukin 4. Okay, so in addition to covaltrum or alum, we do find a lot of interleukin 4, and not much changes in interferon gamma. That suggests, like, this particles is giving some kind of TH2 inflammation. And we also checked by cytokine gene expression for IL-4, IL-13, and IL-10, and we do see increase actually. So our objective was next was, like, it is giving some inflammation. What is the molecular mechanism by which this inflammation is happening? So what molecular mechanism you want to understand, you need to go through the signaling pathway. There are two different type of pathway. One is, like, molecular pattern associated molecular pattern that acts through MID88 and TRAP, and downstream of these is TLR4. We checked in the earlier, I show you that these response happen independent of toll-like receptor 4, but still, like, um, without TLR4, still MID88 trip signal can function. So here to test it, we'll see, like, what is the role of MID88 trip. And if MID88 trip is not working, there's another thing called, like, inflammation pathway. It's called a damage associated signal pathway. There are two pathways. One is pattern associated molecular pattern, that is called PAM, and DAM. DAM acts through the caspase one. So how do you study? How you are going to test this? So basically, you have to have knockout mice. You have to knock these out of the mice, and to those mice, earlier I showed you, like, TLF4 knockout mice. Here we'll do even to see, like, the upstream signal of TLR4 is MID88 trait. So all these knockout mice we will use, and we'll test whether the inflammation is intact or it is gone. Okay, because we have a simple model. We inject the mice, after 48 hours we see by flow cytometer or gene expression, and we'll check. So there are two ways you can do actually to study this. Either you put an antibody to block that pathway, or you put an inhibitor to block the pathway, or you make a knockout. Knockout is the best way to go actually. And so we initially hypothesized that solid microparticle type 2 response is independent of TLR4 signaling, based on my research I published in Journal of Immunology. As you see, like, TLR4 LPS, these are from bacteria. LPS points to the TLR4, then the downstream signaling, there is something called IRF3. Then once IRF3 goes off, like, it can impact on NF kappa B, and then whole cascade of, like, interferon stimulating network comes actually. Like, there are hundred of gene network. These are powerful switches. The moment the LPS will see, it will go and bind to TLR4. So these programming can make cascade of genes. And the TLR4 again acts through the MRD80 trap. And so we'll use we'll knock out, we'll use the mouse with MRD88 and trip. So which will without that, what's happening? So that's what we'll see. The other pathway I say is that DAMP, damage associated molecular patterns. So mostly it works through caspase-1, and there is something called NLRP3. The caspase-1 usually remains in pro-caspase. It becomes caspase-1, and caspase-1 will convert always in our body, pro-IL-1 beta is there. But once the caspase-1 is there, then it'll produce the functional IL-1 beta. And these functional IL-1 beta comes, then all kind of inflammation will happen. So you want to test, maybe DAMP is playing some role. There were some paper with the alum, DAMP might be playing a role. But what we see is different actually. So what we did is that we intraperitoneally inject either wild type mice or MRD trip knockout mice. These mice will not have these particular gene, or caspase-1 knockout mice. This is the PAM signal and this is the DAMP signal. So this is the way you do immunologist signaling to understand which signal might be playing a role. So after 48 hours, we collect the PEC and we'll do flow cytometry and gene expressions to study. So here is the result. This is the flow cytometry. What I explain you, you level with the different markers and you run through the flow cytometer, and this is the analysis part when you do. Okay, I'll explain you. So LY60 and CD11B. If it is positive for both, okay, this is a marker, surface marker. This is a surface marker. Okay, then those are neutrophil. If you have injected with PBS, there is no neutrophil in the peritoneum fluid. But if you inject with cobalt chrome, you see 15.6% or these are neutrophil. There's an inflammation. What's happening with the MID trip knockout mice? Again, neutrophil remains intact. So this is this at least in terms of neutrophil, it is independent. In the caspase-1, there is neutrophil, little goes down, but it is still intact. There is still increase in neutrophil. Same way we look for eosinophil. This is the marker of eosinophil, c-KIT and Siglec-F. And you see over here 1.5% in our in mice peritoneum, and with the cobalt chrome it goes to 13.1, and in amyloid trait remains same. With the caspase-1 knockout, it remains same, and the M2 macrophage, which is very critical, and you see the increase in the wild type mice. So what kind of macrophages are activated by solid microparticle? This is M2 macrophage. The markers are F480 and CD206. So that's remain again intact. Okay, so with this we conclude that it is caspase-1. There is some effect on eosinophil. But let's check the gene expression finally, because rest of the eosinophil and macrophages are really increased. So we wanted to make sure that caspase-1 is responsible for IL-1 beta. So does the knockout mice really IL-1 beta is depleted? So when we give the wild type mice, the plus is wild type mice and minus is knockout mice, and you see with the cobalt chrome there is increase in IL-1 beta, but with the knockout mice there is no IL-1 beta. So without IL-1 beta, still there is increase in innate cells. So what happens to their gene expression? So we will do a quantitative PCR assay. So again, listen to this carefully, because in all you will get a lot of these slides. And so for M2 marker, we'll look for ARP1, YM1, and RELM alpha. For TS2 marker, it is interleukin 4, 5, 13, 33, JAK1, TS2, TH1 marker, IL-12, interferon gamma, and TNF alpha, and DLL4. Okay, so the way you do is that you take the sample, you make the RNA, you convert it into cDNA, and then you put for a specific primer and you do a PCR amplification, and these are talked to fluorescent marker, and with that it will read and it will tell how much genes is there. And you use this is the real-time PCR machine you use to find all this marker. What happens? Okay, there is increase in, these are all the M2 markers. You see there is increase the first two are wild type with or without or with cobalt particle, it remains intact. In MRD trip knockout mice or caspase knockout mice, it looks like genes IL-4, IL-5, IL-13, IL-33, everything is intact. Okay, whatever we see, we didn't see any discrepancy actually. So almost even in the caspase 1 knockout and MRD trip knockout mice, the cytokine response, the inflammatory response is intact. And this is how we concluded that, hey, look, this solid microparticle is causing inflammation as early as 48 hours. And this is independent of MRD-aided TRIP signaling pathway and caspase-1 signaling pathway. So what next? What's happening? We don't know. So one paper came in Nature Medicines wherein they try to understand alum, and again, IL-1 beta could be possibly one of the pathway, they say, because IL-1 beta increase. But other thing they suggested is that even there is something called spleen tyrosine kinase, and downstream of that, there is p38 map kinase, pi3 kinase, whole signaling pathway actually, and there is COX2 pathway, there is a pg2. So yeah. So all these things are responsible for alum. So we try to test actually. So what we do is that first we took sick. If you inhibit sick, what happening to the COCR particle? So there is an inhibitor. As I say, if you don't have knockout mice, you can use inhibitor, the drugs actually. So the drug base 613606 is a known sick inhibitor. So you give -2 days, before 2 days you start giving every day base 613606 to the mice, and day 0 you give the cobalt crumb, and after 2 days you harvest it. Every day you give 1 dose of base 613606, and then you check what's happening to the inflammation. And what we see is that blockade of spleen tyrosine kinase with the bay, I call it bay here. What happens is there is a reduction in the inflammation. You see neutrophil. This is neutrophil CD11 LOS60. My flow cytometric data with cobalt chrome, it goes as high as 27.5. But if you give the bay inhibitor, it goes down. It's a almost five-fold decrease. Same way eosinophil goes down by five-fold, and same way empty macrophages is. This is what graphically I showed that my pro data, that these are statistically significant actually. So that means sick is playing some role with the cobalt crumb. Then even we see they have even impact on empty markers, TS2 marker, and all the other genes network that increased by solid particle are inhibited if you block these pathways. This is another way. So now we got some clue. Hey, look, this sick spleen tyrosine kinase pathways really critical. And so we inhibit with the base 613606 by blocking this sick pathway. It is able to stop the inflammation. So what happening? Okay, downstream of sick, there are a lot of other signaling pathway. We want to know further how sick is acting through which pathway. It can work on BTK, it can work on PI3 kinase, it can work on PLC gamma, there is P38 MAP kinase, it can put on PLA2, and this COX pathway. Aspirin, you use aspirin stop the PG2. That is how it stop. So people have known aspirin can stop some of the TS2 and allergic response, or celecoxib, more COX1 inhibitor. So we tried both the things, but in our case it didn't work. Okay, so I don't have data to show, but it didn't work because this was easy to get aspirin. Celecoxib was costly, but we could get these, and we tested with both aspirin and celecoxib. It didn't have any impact actually in our case. So then we take what's happening, which pathway are happening. So in order to examine that, what we did is that we put the big C and we look for other signaling pathway. We did the phosphorylation. That what happens is sick phosphorylation is very critical for the signaling pathway. So we checked by flow cytometric again, by doing intracellular flow cytometer. that these particular signaling is going off. If you give the bay, sick will inhibit. This is what the drug do. But I'm sorry. In addition, we do see that there is a BTK phosphorylation is also decreased if you put the SIC, because SIC is upstream of BTK. So we check for P38, MAP kinase, PI3 kinase, all the signaling pathway, and we see very less background actually, though the effect was not that progressive. Since we see it's a very good effect, and that to itself, IL-33 is a one of the early cytokine which triggers Th2 inflammation, as I mentioned in previous slide. So we see this bay even inhibit the interleukin 33. So then we wanted to ask questions: does BTK signaling play some role? And BTK is usually, nobody has reported in macrophage what the BTK do actually. People have done this Bruton tyrosine kinase mostly in the B cell. So then we take the inhibitor for BTK signaling pathway, that is called ibrutinib. And what happens if you block the BTK pathway with ibrutinib drugs? So we see that it goes down, all the innate cell goes down. There is decrease in neutrophil, eosinophil, M2 macrophages. There is decrease in Th2 response and M2 macrophages. So these again confirms of BTK is playing a significant role downstream of SIG, and this is something very novel because nobody knows. In Alam, they find SIG, and we find SIG is working in our case. The BTK inhibitor is even much better. So we say that that with the BTK inhibitor, we see that the BTK phosphorylation goes down. There is no impact on SIG because SIG is upstream. Okay, so even without increase in SIG, if the BTK is, you are able to block it, actually it is stopping everything, even the SIG is intact actually. So these confirm, and again with ibrutinib you see the decrease in interleukin 30. Interleukin 33 is a early cytokine which kickstart the type 2 inflammation. Remember this interleukin 33 will come to these. We'll also try to address which macrophage, which cells are producing this interleukin 33 and which is causing inflammation. So BTK, as I say, it is associated with B cell activation. So people want to know, if there is a background B cell. We don't see much increase in B cell or T cell in this response. So we still wanted to make sure that it is B cell independent. So we took a JH knockout mice, which is a B cell knockout mice, and you see it is intact. This is B cell. These mice will have B cell. These mice will not have B cell actually. So everywhere the inflammation is again intact. I'm showing it there is this is a neutrophil, eosinophil, and M2 macrophage. They are intact. The cytokine pattern that I showed you and explained you, they are intact. So we confirm from these that this is independent of B cell. So that means what the BTK signaling pathway is going on and it is impacting interleukin 33. So that's our next question. Then ibrutinib is also some people say it can act on some of the T cell actually. So is it independent of T cell? So for that we'll use the we didn't get the mice actually, so we used the antibody actually. We depleted the CD4 T cell and we tested that it is independent of even T cell. So as I say, B cell actually in the JH knockout mice, this is how you check. CD19 is the marker of the B cell. So this is the PBS or cobalt chrome. There is not much increase in B cell, but in the knockout mice there is no B cell. So our knockout mice was perfect actually. So then we use the GK 1.5, which is an antibody against the. It will deplete all the T cell if you give this antibody to the mice. Okay, and we see the response remain intact actually. Again, there is intact neutrophil, intact eosinophil, and intact M2, and so the gene expression. So we conclude that this inflammation is independent of CD4 T cell and independent of B cell. So then what remains is our neutrophil, eosinophil, and macrophage. Out of that, someone is playing a role. Okay, so then we also, to confirm, we used BTK inhibitor, but we wanted to use the SCID mice, which are known for those are BTK mutant mice actually. So in the mutant mice, what's happening? You see wild type mice, there is increase, it goes down. Here also the eosinophil goes down, the M2 macrophage also goes down. You check this one and this one. This is just with PBS. This is with cobalt chrome. Okay, this is again with PBS and cobalt chrome. So with the mutant mice, again we confirm. We first tested with the BTK inhibitor, ibrutinib, and now we confirm that, like, if you take a knockout mice, because always scientists wants to test in a knockout mice what's happening actually. Okay, because drugs can have a different impact. It can act on different pathway. So in order to solidify the result, scientific investigation, you have to use the knockout mice, and we see that, like, this is a BTK dependent pathway by using this knockout mice. So then let me okay, so then um since we see, like, interleukin 33 is going down, what happens in the BTK knockout mice? If you add, like, the interleukin 33 exogenously, does it again kickstart the type 2 inflammation? So we add, like, to these are all, like, BTK knockout these past two are, like, this is wild mice, and this is the BTK knockout mice where there is no neutrophil or eosinophil or antimicrophage. But once you give interleukin 3 exogenously, we give recombinant protein interleukin 33, and you see, like, again it come back. So this is interleukin 33 dependent. So these results, like, confirm that again, like, if you exogenously free interleukin 33 is very critical. Okay, so then we want to know what the immune cell is producing interleukin 33. Again, you use flow, but this is a different type of flow wherein, like, you can sort each cell. Hey, this is macrophage. I need macrophage in this tube. I need eosinophil in this tube. I need neutrophil in another tube, and T cell and B cell in another tube. And you can collect those cells, and you do the gene expression from that. And we look for IL-4, IL-5, IL-13, IL-33, IL-1 beta, all the markers we see, even interleukin 33. And what we see is that interleukin 33 is mostly in the macrophage. So macrophage is one of the major cells. So macrophage is the major producer of interleukin 33 in this model of solid particle mediated sterile inflammation. This is again another new investigation, and how macrophage is producing interleukin 33 was another question which was very striking, and we try to address that also. So then again, like, in order to confirm, like, blockade of IL-33 really, like, stop these things, and IL-33 is a major player for these kind of Th2 inflammation. What we do is that we block the IL-33 receptor by ST2-FC. ST2-FC will block the receptor where IL-33 goes and binds. And we see again, if you block the IL-33 response, you see the decrease in inflammation. So this again conclude that IL-33 is really critical, and these macrophages are producing interleukin 33. So then this was all in peritoneal model. Since it was an orthopedic model, we wanted to test what's happening in the knee model. So we try to address this by injecting mice intraarticularly. So as you see, like, it's so difficult. The mice knee is over here, and this is the synovium. Exactly, I had 3 or 4 orthopedicians, MD student there working, and particularly Mark Palmer is my favorite. He's the one. He's a very good surgeon, and he will be able to techniques, like, wherein he could inject, because this is a very small space in mice. So the microparticle will exactly go over here actually. So he tried with almost, like, many mice, like, more than 500 or so, and he practiced this technique, and he made it. Once it worked, then we tested what's happening actually. So we check, like, initially you see, like, this is a PBS mice, and this is with cobalt chrome, and there is inflammation in synovium, and we check the different gene expression by extracting synovium, separating the synovium fluid, and getting the cells and doing the gene expression from there. And this is an inflammation score he used to do by measuring the size of the inflammation, and he has some scoring way actually. Uh, then I was interested in what happens if you give the SIC inhibitor or BAY inhibitor. Is it stopping the inflammation in the knee actually? As you see in the PBS, this is the synovium portion, and if you give the cobalt chrome particle, you see, like, there is inflammation. And if you give bay, the inflammation is gone. And if you give ibrutinib, again the inflammation is gone. Here, look at the inflammation. And this is inflammation score with a cobalt chrome particle. There is increase in inflammation. With the SYK inhibitor, it goes down. Again, with the BTK inhibitor, again it goes down drastically actually, and these are statistically significant. And we also check for different cells actually in the synovium, and we do see with these inhibitor, even in the synovium, not only in the peritoneum, even these TH2 response happens in the synovium, and these inhibitor are working. So then okay, these are some of the orthopedic stuff he does, bone area, he does trabecular number, and different way actually. This is beyond my knowledge, I'm an immunologist actually. But basically he see CSCR can cause these differences. And also he check for additional pathology. And he says cartilage thickness, you see, or the cartilage thickness go down. You see this red, more red is the thickness of cartilage is really good, and it goes down. So all these things I could do because some of the orthopedicians were really working with me, and they're very interested in this research. And particularly Mark Palmer, Bonnie, Jay Moore, all are now good orthopedicians in many different hospital right now. So we also wanted to check what happens in the human patient. Do we see increase in M2 macrophage marker? Because M2 macrophage is what increasing with CSCR in mice. So what's happening in humans? So from mice, you have to go and see whether the biomarker, what you see in the mice, is it there in the human? So then they have, we are attached to some of the surgery curves. So they used to get there, and these people are involved there. So they used to get, and they used to do all the study. And as you see over here, this is a polyethylene particle. We didn't get a COCR particle, good one actually. Mostly all the cells are dead, and it was so difficult to stain actually. But these are all we do frozen sections actually. Means they'll get from the sample from the hospital, and it will come in liquid nitrogen, and it will immediately make a OCT sections, and they'll make the sections, and we'll see. First we'll do a HNE staining, and in HNE staining you see this is polyethylene particle, and it is causing inflammation. And we check for CD60 and CD206 for the M2 macrophages, and you see there is increase in these macrophages. And we check for ionos. There is not much changes in the ionos. CD68 and CD163 is another marker actually. You see increase in mostly in the human pathology. You check for M2 macrophage, CD60 and CD163. And we check CD206 also, and we check also other different marker of the M2 macrophage. We see increase in interleukin 3, ARC1, MRC1, chitinase 1. These are all the naming are different. Interleukin 33 is increased, and there is not much changes in IL-12 or INOS, which is M1 macrophage. I told you before. So what's happening is that here, so even solid particle mediated inflammation, that is increased in the M2 macrophage, and there is increase in interleukin 3, there is increase in markers of M2 macrophage. Okay, again in human, MRC1, CHIT1, and ARC1 are the marker of the M2 macrophages. I'll show you a video actually. What we did is that we purify the macrophage from the peritoneal macrophage. We tried with bone marrow, it doesn't do that good. Okay, we take the peritoneal macrophages, we separate it, we confirm by flow cytometry that these are really truly macrophages. And you'll see in the video the black one, what you see is that this is these are all COCR particle, so you see the red mark. What I'll do, what will happen, you'll see how they'll engulf the macrophages, and what happens, it will die actually. So you'll see all the thing. I'll put 2 times. This is a very short video. Can you see? Look at that. So here, like in the white symbol, what you're seeing, like it took macrophage, and what happens? This is just like a bullet. This is not bacteria or fungi or this thing that macrophage can gulp it and start presenting the antigen. These are solid particle. Even they produce whatever enzyme, this cobalt chrome, this solid particle metal is not going to go anything. The macrophage dies in the process. What happens? Macrophage dies. It try to kill it, and there are some macrophages, they cannot even they try to assemble around. You see over here, they assemble around. This is a bigger particle, they cannot go, they keep on. So a lot of macrophages are dying. So there is a dead macrophage around here, and they'll try to surround. And we have seen this video for 72 hours, and we have so many slides, and we try to see how they try to make a net kind of structure. Their movement are different, and as early as 30 minutes macrophages start take it, and by 24 hour you'll see different cell death will go and macrophage death. And we believe this macrophage death perhaps produce the interleukin 33 with the necrosis of macrophage. And I'll put it again. You see macrophage is taken here, and you see immediately it dies, and again dead macrophage is taken by another macrophage. So basically some kind of things what we immunologically call is, it's called frustrated phagocytosis. Macrophage jobs, they are scavenger. They'll see they are any garbage they'll see, they'll try to pick it. But these are they eat it actually. They eat it and they try to digest, and they present it so that T cell and B cell can go and do their second defense mechanism to protect us. So here what's happening is that this is called frustrated phagocytosis. They try to phagocytosis, they try to eat the metal particles, and what happens? They die. And some of the metal particles are bigger in size. They surround and they keep on dying. And this process is called frustrated phagocytosis. And perhaps this might be producing interleukin 33. In fact, we have done some experiment with the IL-33 reporter mice, which I'm unable to show it in this presentation, that this is what happens. So then we also we took the macrophage, we put the cobalt chrome, you put cytocolsin D actually, that will reduce actually the actin and everything, and what happens. And then we put the necrosis or apoptosis inhibitor, and what happens to interleukin 33? What we see is that if you block the necrotic pathway, you see the reduction in interleukin 33. And this is what you see we count how much phagocytosis happens actually, how much metal particles are there inside. And with cytokinin D, it's a phagocytic inhibitor. And what happens to the interleukin okay. So with this we showed that if you use a necrosis or cell death inhibitor, it basically stopped producing interleukin 33, and that's statistically significant. And then we earlier showed you caspase 1 independent IL-1 beta is produced, but independent of caspase 1, this response you see intact interleukin 33. And this is what it is. So basically we proved that it's a cell death pathway, and death of macrophage is so critical, particularly in this model of inflammation, and that produces interleukin 33. And once interleukin 33 comes, it kickstarts the whole type 2 inflammation. So again I'll probably summarize. My first part is taking so much time. So the mechanism, in order to understand what we think is, cobalt chrome particle comes, and this is the macrophage cell membrane. So the moment it touch, independent of structural composition, the moment it touch actually the lipid bilayer, this gives signal and stick get lighted up, phosphorylation. And this downstream there is BTK phosphorylation, and with these BTK phosphorylation happen, it produces interleukin 33. And by this time this is dead, and interleukin 33 is released from the macrophage. And in this response, basically what happens? What are all the immune cells increase? Neutrophil, eosinophil, and M2 macrophage. And what are the Th2 type inflammatory markers? Interleukin 4, interleukin 5, interleukin 33, and interleukin 33. What are the M2 macrophage marker? ARC1, YM1, and RELM alpha. So based on these, we see that CSCR particle increase a type 2 inflammation, leading to cartilage and bone damage. And you guys are using some of the some of the, you guys are using some of the dental material that might be also like causing some kind of inflammation if the particles are releasing. However, like in the dental, like it's a filled with oral microbiome, so that can also, LPS can also cause some kind of other, like with the co-infection, like it can cause different thing. With these, like I acknowledge my all the colleagues who works with me, and with these, like I'll ask them if you have any questions on the past part, you can ask me.
Sir, as of now, there is no questions. Maybe at the end of the second session, we'll see for the questions and then we'll
Okay.
answer it.
Okay. So yeah, I think we are running out of time. I have to finish in 10 minutes, actually, 10 more minutes. I think. Yeah, other thing I work right now, my major thing is that I work on CAR T cells and cancer therapy. So basically, CAR T cells is again, this is becoming these days, like CAR T cells are living drugs. They can what they can do is that, like you can target different tumors, and the way you do is that, like you basically, like there will be a lentiviral vector, and you introduce particular chimeric antigen receptor. And what is chimeric antigen receptor? It has got a antibody. It's called SCAB because you just give a heavy chain and light chain, and this has got a hinge region, and there are some co-stimulatory domain and signaling domain, actually. And CD3, there are different signaling, co-signaling domain like CD3 zeta, CD22, CD28, 41BB, OX40, and even for NK cell, different type of co-signaling like DAF10, DAF11, NKG2D. So different components are there. So basically, you want to introduce these to T cell. So you get the T cell and you want to introduce. How do you do this? You use a lentiviral vector. Lentiviral vectors are nothing but just like courier. You want to courier, send some gifts to your dad or mom. How do you do? You courier them or send it by post. So just like that. So you package it. You put in a box. So basically, like virus has a envelope protein. It'll go, and it has a integrating gene, actually. So this gene will be come and it will integrate into the CAR T cell. Then once it happens, this will start getting expression over here, and the virus is gone. Once it integrated, once it has happened, this one does the role and how it works on the uh, killing cancer cells. So there are like these CAR T cells are, as I say, like these are living drugs. These are right now, it is our personalized medicines, and there are initially, like the first drug, FDA approved drug, came as Chimerio. It is produced by Novartis. Then later on, like there is Yescarta, Tecartus, Breyanzi, Evicma, Carvicti. So these are all, like remember, like what are all the, like these are the first two FDA approved CAR T cell. And in fact, like here the target antigen is CD19, and they target for B cell lymphoma. Mostly these are for B cell lymphoma, and they're all targeting for some B cell markers like CD19. uh, Here for Chimera, they use the, like mouse antibody, the SCAB portion, the antibody portion from mouse antibody. They use that gene, they put to express in the human T cells, and that works so well. This was with these success. Then Yescarta, what they do is that they just did little bit changes. Actually, they use the human CD19 over here, and then like then there are other things, little here and change. Then BCMA is another marker, actually. There are some people have used. So but the good thing is that there are 6 FDA approved CAR T cell therapy, and it is in high demand because the cancers are really, like cancer can cause death, and with these, like a lot of people are able to revive from their cancer. So what happens in the CAR T cell? So basically, like you express uh, antibody against a particular target. The cancer will be expressing different target. So you wanted to, your T cell to go and bind there. In order to do that, you produce the heavy chain and light chains, actually. That is called the SCAB. So you either take mouse SAB or you humanize the mouse SAB. You change it just genetically. You can change. You can make it humanized. Okay, just the amino acids and proteins should be same actually. Or you take the pull directly from human SCAB actually. If it has been characterized and known sequence are available, you can clone that. And or you take only the heavy chain variable region actually, or even no need of antibody. You can take tumor receptor ligand instead of SCAB, and you can express those things in the T cell, and those are called chimeric antigen receptor. So basically, the way it works, this is the flow diagram I want you to explain. What you do is that, first thing you do is that once cancer has been once they don't get any response to any drugs or nothing is working, then they go for the last option is CAR T cell options, because this is very expensive. This is personalized medicine. Earlier it was costing $1 million per patient, because making these CAR T cells, it's looks very simple. I'll explain you, but there are a lot of complexity in between. Okay, these days it's available at $0.5 million, 250,000 to 500,000 per treatment. And so, simple thing, what you do is that patient comes, it gives you the blood, okay, around 250 ml or 200 ml, and then you separate the T cell from the thing by, there are different techniques actually, separation method. I showed you in one of my previous slide how I separated the T cell from mice. So same way you separate with using different beads, actually magnetic beads, and then you insert the CAR. CAR is what? Chimeric antigen receptor. As I told you, here mostly B cell lymphoma. So the CD19 antibody will be you put package into lentiviral. Lentiviral are nothing but they are just like a courier company. So that will go inside and it will express. Once it come inside, the once you transduce with the lentiviral, that is just like a courier. It will introduce the gene of your interest, whatever you want to express in the T cell, and it will start expressing this antibody. These antibody are against tumor cells. Okay, so the next thing what you do is that grow millions of cells actually, because you give 1 million cells per kilogram, in that range. So then once it grows, it takes around 3 weeks actually to grow. So the timing is very critical. A lot of people are now trying to tell, the time you collect the blood, it takes almost 4 weeks, 3 weeks to 4 weeks to go to the, again, it can be made personalized, and it can be given to the patient. So that's the reason it is called living drug, because this is a different type of drug, living drug. You take the patient's own blood and you modify genetically by expressing chimeric antigen receptor, and this has got antibody, and these antibody will target the tumor cells, exactly what is happening. So once you infuse to the patient, immediately it will go near the cancer cell, because this antibody will, there are so many number of antibody, there are different way how many antibody you want to express in the T cell. All these things can be controlled by doing the cloning and exactly designing. So designing a CAR is so critical. How you design, how many SCAB you want to express, so that exactly you are able to reach to the cancer and it's able to fight, and also you have stimulatory, co-stimulatory molecule signal, so that the T cell will start producing some of the cancer killing agent actually, and then cancer cell will gone. So this is what is the diagram. So I will tell you once again. So first we remove the blood, then we purify the T cell, then we do the genetic modification. We introduce the gene of interest, here for example CD19, and then we say that taste that it express that particular antibody that we want to express, and then we grow millions of cells, and then we infuse to the patient. Then we see once it goes inside, whether it fights with the cancer, and whether cancer is gone. For that also you do monitor. So this is how this is the chart, a flow diagram of the CAR T cell therapy. It takes a month actually to do these things, and since each one is from individual cells, um, it's laborious, and a lot of high techniques are required. And in order to give this living drug, FDA needs a lot of testing are required that these are safe actually. So people are trying to do, like, to cut down the price, like, how about, like, if it's available in $2,000 or $1,000, it is not possible taking from one. But however, if you take from one, some any volunteer actually donating, and if you're doing modifying and making some universal CAR T cell which will not be rejected by a different patient, then probably that will be the best. And how to do that? So, like, there are the different generation of the CAR T cells actually. First generation, second generation, third generation. First generation was very simple. You put the antibody, like, say for example CD19, and you put CD3 zeta signal. Okay, this is the activation domain. It will activate, and once the CD3 zeta is there, it will produce all the cytokines, whatever is required to kill the cancer cell. In the second generation, they put one co-stimulant signal, like, say for example CD28. Okay, in the third generation, they put two stimulatory. Like, we use, mostly people use 4-1BB ligand and CD28, and this is a CD3 zeta. So the car will be called, like, hey, I made the car construct with CD19, 4-1BB, CD3 zeta, and CD28 car T cell. This is what mostly people are using. And there are fourth generation wherein you can give a particular cytokine and you can switch on. You can have suicide gene. If you see, like, there is some problem with the CAR T cell, or it is growing heavily, you want to kill them or you want to shut down them, you can introduce suicide gene, you can silence them, or you can do. And there are a lot of modification, like, these T cells, like, they can even, like, induce, or they can adjust, and they can die in order to make this living drug durability, how long it will stay. You can even knock out some of the things, like, PD1. There are other genes actually you can knock out. Then these CAR T cell also produce cytokine release syndrome. So, like, so different cytokines you can delete. So these are all things that are going on right now actually. So one of the major thing is that, in order to map the how many copy number of the CAR T cell, particularly the antibody that your T cell will express, so that it will go and bind to the antibody, is very critical. So the way you design is the promoter. What is promoter? Promoter is a region upstream of where the expression start, wherein, like, RNA polymerase binds and do the expression. So there are strong promoter, there are weak promoter. Here, say for example, like, EF1, there is CMB promoter, there's a PGK promoter. There are different promoter, and it has been tested. So I will summarize here, like, so this is a heat map. Okay, basically the heat map has a scale from 0 to 10. If it is yellow, 10 means it's really very good. And here the function, LB titer, this is the lentiviral titer. How much tighter? If you do with EF1 promoter, your car construct, like, if you are expressing with the EF1 promoter, these are all the grade. Most of them are yellow. CMB promoter. It is CMB promoter is a very well-known promoter. Most of the expression vector has actually, but it is weaker, little weaker actually. It is in the middle actually. Then there's a HPGK. There is RBB, SA. There's also yellow some of the person. So depending on the length of the, again, length of the transcript actually, what is the size of the transcript? Depending on that, you design which promoter to do, so that you are making a very good CAR T cell. And that is where, like, all the money and business actually. So people try different promoter. This is just 4 examples are given. People try, like, trying different, like, 20, 40 different promoters, and trying to address, like, where, what is the new things they can insert, so that they can make a thing. So again, like, for the student, what are all the promoter? What the promoter does? Promoter basically helps in the transcription of the gene of your interest. Okay, so EF1 is one of the best promoter, and CMB is another promoter, and so EF1 alpha promoter is one of the strong promoter based on the scaling actually. So then the question is that, like, how to make a universal CAR T cell, so that it will be, like, affordable, like, $500 or $100 per cell. So if you make a large quantity and keep in the store in the -80, and whenever whoever needs, you give that drugs, then it is possible. How do you do that? This is from a paper from Science in 2000. Carl June was the doctor, like, who is the first one who gave the CAR T cell. This is one of the paper from him. So what they did is that they tried to delete actually the TCR, T cell receptor, T cell receptor. So if you receive by they remove the TCRC and the track B and track C, that is 2% of the TCR, T cell receptor. So with that, you can make it universal. And they try to also PD1, they try to knock out actually the PD1 by using the CRISPR and Cas9 technology. The way it works is that, you basically in your genome sequence, you see what is the PALM sequence. PALM is nothing but NGN. G is guanosines, N is any nucleotide. So based on that, you design a guide RNA because it will come and cut it over here actually based on the PALM sequence. So you design for PD1, you design for a TCR, and by removing the TCR or even class 1 HLA, you can make it universal. But this is still a trial is going on. So there are different genome editing tool people are trying. CRISPR-Cas9 method, there is a ZFN method, zinc finger nuclease, there is a Talon method. These are the methods for genome engineering. The problem is that it's so easy to do the CRISPR-Cas9, but what happens is that when you are introducing, there is most of the time there is off-target effect. What is off-target effect? Like say for example, if it cuts here, but still one or two nucleotide always goes either deleted or that is called indel. Either it is an insertion or deletion, and that can impact the downstream gene of things. So then that could cause again cancer. That could cause a lot of problem actually. So that's the reason, once you do these kind of genome engineering, you need to check that every other 30,000 human gene, all these things are intact, and to do these and make it certified that this is safe and this may not cause any autoinflammation or any problem. You need to that's the reason, this is where a lot of research is going on right now. So this is a future design of the CAR T cells. So use CRISPR or Cas9 method, and you make one of the problem with the CAR is that you get the cytokine release syndrome, wherein you get the GM-CSF, interleukin 6, interleukin 1. So probably you can delete those, or you can produce even some of the antibodies, so that those cytokines when it comes, it can neutralize actually, and you can make the universal TCR knockout, and you can make the universal core. And once that will happen, probably it will be cheaper. So here is an example actually, just to go briefly, this is a cassette. So basically this is the antibody portion, then it linker, then this is another, you can put even dual antibody actually, so that it will target better actually. Instead of single antibody, you can express dual antibody, CD19 here in, for example, CD19 and CD22. Then you put the 4-1BB co-stimulator signaling and CD3 zeta. So when you design these, all these components matter. What CD19 SCAB also, there are many are available actually. Which one you are using, which linker you are using, whether it hinge, what is the size, what is the length, how much is the length, you see how it is hanging, whether the length you can certain by changing different linkers. All these things matter. 4-1BB, CD3 zeta. So all these things matter. Some places they use dark 10. So all these things are so critical. So major thing is that antigen escape. Sometimes you have to see the antigen what you're designing is really very good expressed in the tumor cell, and the tumor doesn't escape the antigen. So it has to be particularly for B cell lymphoma where it is successful, because CD19, CD20 these are always expressed actually. Then you use with the CAR T cell, they target, then up target on target up tumor. That means if you use CD19, CD19 is also a marker of the B cell. So in addition to depleting, killing all the tumor cells, they'll also kill all your B cells, normal B cells. Okay. So that's something. So that's the reason you need to see how much thing you have to do, and it is critical how powerful you want to make. If you express more, it'll be a more powerful, but it may not be good. It'll even deplete all your B cell. Then If you get any kind of infection, it will be problem. You won't be able to make antibody. Okay, a lot of people take CAR T cell in the SARS-CoV-2 time, COVID time. We see like their response is very poor. So we need to do then plasma transfer so that they can be protected with the SARS-CoV-2 disease. Then trafficking, and once it goes, there are a lot of immune suppressor cell, like myeloid suppressor cell, then T regulatory cell. They can also suppress the effect. And then one of the major problems with the CAR T cell is that it releases a lot of cytokines like IL-1 B, IL-6, and TNF alpha. But there are drugs, and this is just like fever actually happens, and one can control it actually. So in order to design like the CAR T cell, basically like this is the lentiviral construct. So basically, as I say, like you need to put in a delivery package actually to introduce to the cell. So you basically put four plasmid. One is your CAR gene, one is the envelope protein for the virus, one is GOC and polymerase, so that it will be able to amplify this, and one is reverse transcriptase. So all these four plasmid you transfect to a cell line actually, and then they produce this lentivirus. Then they have this gene of interest. So this is packed, already packed actually. This is the courier. The way you make the package better, it'll go. Then you transduce to T cell or NK cell. Just put this virus. This will go and change your T cell, and then you expand. You expand to different cytokines. So how you expand actually depends. There are also people do like TIL, tumor infiltrated lymphocyte. They get and they try to make the core of the TIL. They are cancer specific T cells, so they go more better. And there are different way of like expansion of these CAR T cells is very critical. I will skip these slides. Actually, this will be too much, I think. But to tell very briefly, like you want to check like different markers in the T cells, so that they have like effector, whether they have a memory-like function or effector function, and you want to have a CAR T cell which is a memory-like function, then which cytokines you want to expand, and whether you can expand the T cell within a 1 week instead of 3 weeks to get like 300 to 500 million cells, so that you can inject to the patient. So these are all the things. And here is an example, like this is one of the things. You look at the thing after a 6 week of thing, this has drastically gone down actually. By and here is another example actually. The tumor goes down drastically like after 6 weeks. And one of the things is that when you are trying to develop actually a lot of things you need to do. You need to make sure like the purity, there is no endotoxin. You need to check identity. You have to do next generation sequencing. You have to do like 40 to 50 markers with flow cytometry. You have to do potency assay that the tumor that you're targeting, it is really killing those cells by doing video microscopy, and check that. Then you check sterility testing, like it is without mycoplasma, without fungi, without yeast, and you do all the things in sterile environment. You make sure like the products are really safe and good to go actually. So different analytical strategy basically you use actually. You use flow cytometry, use digital here to check the copy number. You titre the lentiviral particle. You do different functional assay, like what kind of cytokines they produce, or the chimeric cell, what kind of phenotype, whether there is antibody dependent cellular cytotoxicity. Then you use the next generation sequencing to see that all the genes are intact actually in the T cell. You do nanolife video microscopy. You see their telomere length. You do in the mice in vivo function, and as I explained, sterility testing, that it is not contaminated with any bacteria, virus, or fungi. So this is all like to give you a very short introduction to the CAR T cell, and I'd like to say thank you so much. Probably I took a little bit more of your time.
Question, sir.
Yeah, is there any
Yes, sir.
question?
Thank you very much for your wonderful lecture. It was in detail about the CAR T cells, sir, and the treatment modalities, in-depth mechanism, and all those. It was really interesting, sir. Most advanced technologies to our students to know. Coming to the question, sir, um, Angel had asked, is there any adverse effect of chemo treated CAR T cells, sir?
Yeah, there are also the concept of antibody drug conjugates, so you can have antibody drug conjugates so that antibody will go tumor has expressing different antigens or different proteins on their surface, the tumor. So you basically the SCAB or antibody what you said, that is antibody drug conjugate. So you can target that. So in addition to the T cell producing releasing cytokines and cytotoxic modality, even there will be antibody drug conjugates, and once antibody bind, the drug become active, and even the drug works actually. So there are different ways. Particularly, the major problem is that solid cancer, because solid cancer are bigger in size. So there are many ways of doing. T cell is one of the things people are trying with NK cell, and I showed you the video of macrophage how it even takes the cobalt chrome. So same way, people are trying. They call it macro core actually. A lot of people are trying for macrophage core. So probably for solid tumor, which is the one of the major difficult thing. Probably macrophage core can go and the idea of the CAR T cell is that you are designing basically the antibody binds to the tumor, and the moment it binds, there is a stimulation. CD3 zeta is there, that will activate, and just like a putting a bomb, it'll start releasing all the cytotoxic substance, and tumor will start depleting. But when it is a solid tumor, these are being one by one, making it fragmented, and so then in that case, there are you need to probably do drug and antibody drug conjugates, and you have to try different markers, different antibody, and different treatment together actually.
Sir, our Ramya wants to know if there is any age restriction for this CAR T cell therapy, sir.
No. In fact, the first the cortisol therapy was given to Emily. Her name is Emily. She was 11 or 12-year-old actually with the B cell lymphoma, and she was the first successful. It was given in NIH, and she comes to all the CAR T conferences these days. She's very young now, and she was given just one dose, and surprisingly, the CAR T cell remained even after 10 years. Some of the CAR T cell are still there, and she never got actually. The first case is somehow fortunately because very good doctors were there. They see this all the cytokine release syndrome and all those things. They could really stop it and make it go and work actually, and that was the first successful. And most of the people these days, they when nothing is working actually, even old age people actually, they're getting CAR T cells, and this is becoming much and much demand. That's the reason, this is just like mushroom. The industry are going up, and there is always technically how they can compete, how they can reduce the price so that there is more demand and more people can get it. Initially, it was costing $1 million, and most of the patients. But if you see, they're older patients.
Sir, how do we select safer antigen, sir?
Can you repeat it again?
How do we select safer antigen, sir? Question from
Oh,
our
yeah,
student.
that's very good questions. Actually, that's the major thing. Okay, in the B cell lymphoma, the lymphoma is caused by B cell. Okay, B cell marker are no. It's CD19, CD20, CD22, or B cell maturation antigen, BCMA, and there are many other things. Okay, they're very well known. Okay, so for tumor, each tumor can have a different. So these days, people what they're doing is that they take a biopsy of tumor, they do the next generation sequencing. So will you will know all the 30,000 genes, which all are very highly expressed. Okay, say for example, you find the best 10 candidates actually. Okay, from the next generation. Hey, these 10 candidates are high. Then you check for which all could be in the surface and which one will be inside actually. So you want to target which are on the tumor surface so that this CAR T cell can go and there. So biomarkers is every company has a biomarker discovery group actually. So they produce different biomarkers, and when it calls personalized, some people have can even they try to these days different new technology are coming wherein we can do genome engineering, like they do library of different markers, and it will be expressed, and it will go, and for sure, like it will go to the target site, and it will be able to result the cure.
Sir, is RT therapy approved in India or other approved countries, sir?
I didn't get it properly. What you are asking is probably like is it approved in only in
Our
US,
country,
other countries?
only in US or even in other countries, our
No,
students wants to know. If
definitely
yes,
this is there in different country also, and slowly they're expanding. The models are becoming different, like whether they can make these like more automated making, like wherein like and with a simple pod, like pod like structure. There are different models, different company are following, and their expansion, how they can reach to the. Right now it is mostly in the developed country actually, but it is slowly like expanding, and because the model is like you need to get the patient blood, and immediately you need to make it actually changes this, and you need to expand, and you do that. So the model is also like they can freeze storing these. If you do cold storage actually also, like the function goes down actually. So these are all challenges. So there are many company has different patterns on how they store and how they transport um, uh, to a different country. But it is more like they are right now whatever therapy are there mostly local actually, and very personalized way actually right now. But it is slowly coming up actually. There are definite company are having model wherein like they want to probably attach to different or each hospital. Say for example, they have a facility wherein like they can do something like this, and it can work out. You know, it's just the protocol and model what they are following up.
yes, sir. Next question is how to overcome antigen loss relapse in hematological malignancies.
Yeah, so like that's a very good question actually. Like sometimes like uh, the antigen loss can be like but it can happen, or because some of the tumor or like antigens or like it express in the membrane, and sometimes it goes and hide actually. Even if you treat with certain drugs actually, some of the tumor markers what you'll be seeing expression that may get masked actually. That's quite tricky actually. That's the reason like a lot of characterization is required actually, like with the tumor biopsy you try to see like which all markers are really there on the surface, and even with a particular treatment, you check like whether any masking is happening or it is still there.
Sir, thank you, sir. Thank you again.
Yeah, thank you so much, and
Thank
for
you.
all the student, dental college student, I wish you all the best. And
Thank you.
some of the part of my talk you may not have able to follow it actually, but
Yes, sir.
this is a way
But
like always keep your hope high. And here, like at North America, mostly like doctors, they see the patient and they want to do research also. The best of it, they want to do research and they want to see the patient, and which is somehow I didn't see what is the status now back in India. I don't know, but it is always good to do like have some kind of research mind and asking questions when you are failed in treating patient. Always think of a research and always keep option. Talk with the scientists and try to find it. Okay. I hope with this forum you learn something, and I wish you all the best. And thank you to all the organizers.
thank you, sir. Few words from Ramya, sir.